Thank you very much. It's my honor to be here. I appreciate the opportunity to speak on this occasion. So the subject I want to talk about is a new approach to broadly protective vaccines. Sometimes we call low sugar universal vaccine. How do I do this?
The right-hand side.
Right?
You're upside down.
Oh, upside down.
Yes.
So all living cells have a sugar coat on the surface, and on the right-hand side is a typical blood cells, and you see the furry part on the surface are carbohydrates. Sometimes we call sugars. It's called glycocalyx. But we don't know much about the role of sugars on the surface of cells. And on the left-hand side is a COVID virus. And the orange dots are the sugars. So the sugars actually are made by the host, not the virus. The virus cannot make sugar. They depend on the host glycosylation machinery to complete the life cycle. And these sugars usually are there for two major purposes. One is to facilitate the infection, and the other is to escape immune response. So if you look at the evolution of SARS-CoV-2 from 2019 up to now, you can see that there are a lot of mutations going on. And you probably still remember in 2021, in October, there are about 5 major variants of concern, including the alpha, beta, gamma, delta. And then after 1 month, it's all about delta, more than 98%. And after another month, delta disappear, and Omicron start to come out. And in January, the dominant strain is Omicron, and now it's all Omicron subvariants. So you can see that the speed of mutation is much, much faster than the speed of making a vaccine. Even mRNA vaccine cannot catch up the rate of mutation. So this tells us we need to do something to contain the pandemic. And that is why universal vaccine concept came out. So we look at the sequence released by GISAID since 2019. There are about 15 million spike protein sequences, and we try to find out which epitopes are highly conserved. So from this, you can see that there are about 18 there, but 17 out of the 18 are covered by sugars. So in other words, our immune system usually don't see them. And then if you look at that in more details, the most highly conserved epitopes are located in the stem region, very similar to the influenza virus and the HIV. And in the S2 domain, that is the C-terminal domain, there are about 11, and which are also highly conserved. And out of these 11 epitopes, 7 are located in the stem. So this will be the target of universal vaccine development. And what we did first is just to remove the sugar. But if you remove the sugar completely, the protein will fall apart. It's a trimer, so you need to keep the trimeric structure intact so the B cell can recognize the shape of the protein. And so what we did is to trim the sugar down to monosaccharide and to make sure that the protein is still in its intact three-dimensional structures. So on the left-hand side, you can see that it's the fully glycosylated spike protein, and the blue colors are the sugars there. And on the right-hand side are the monoglycosylated spike protein. You can see that the stem region is fully exposed. So what happened to this? So we express the spike protein from two different cells. One is the so-called BEAS2B. That's the non-epithelial cell, which is the primary site of infection. So you can see that the spike protein has a blue color and a green color there. And the blue color are the sugar we call complex type of sugars. And the green color is a high mannose type. If we remove the sialic acid from the blue color, then the infection rate, the infectivity is reduced by about 4-fold. Now, if we trim the sugar down to monosaccharide, the binding to the receptor, the ACE2 receptor, is reduced by about 10-fold. So this has some implication about safety of the vaccine, because you don't want the vaccine to go to every cell through binding to the ACE2 receptor. So these are, in terms of safety, I think we are in the right direction. Now, what about immune response? So we immunize the fully glycosylated spike protein versus the monoglycosylated one. And you can see that the results shown here. The red colors are the monoglycosylated one. You can see that the IgG titer to the target is higher, and the neutralization activity is better, and it also induces CD8 T cell, memory CD8 T cell response. And I think that is very important from a vaccination point of view. And in the challenge study, you can also see that it's almost fully protective against the vaccination strain and also the mutants. And then we look at the neutralization against other mutants. You can see that it's also much better than the fully glycosylated vaccines. Then we want to know more about why it becomes broadly protective. So we use the single B cell technology, and then we use a fully glycosylated spike protein to sort it out, to sort out the single B cell which bind to the fully glycosylated spike protein, and then we take out the antibody gene, and particularly the FAB gene, and then combine that with the humanized FC constant heavy chain gene there, and to construct the monoclonal antibody library, and then screen for the effective antibody which neutralize the virus. And we found that it's very different. The antibodies induced by the monoglycosylated vaccine is quite different from the fully glycosylated. The antibodies from the monoglycosylated vaccine are much broader, are much more diverse, and you can see that here is one example. We isolate one which target the glycosylated region there, and it binds to the fully glycosylated spike protein at picomolar binding constant, and the neutralization activity is also in the picomolar range against the virus strain and also other mutants. So this tells us that if you use a monoglycosylated spike protein as a vaccine, it will be able to induce the antibody which can neutralize not only the vaccination strain, but also many, many other mutants. And the sugar there will inhibit the induction of the antibody. But if we remove the sugar, then the immune response is much broader and stronger and induce the antibody, which can actually recognize the glycosylation epitope. And I think the way it works is the antibody will bind from the other side and then push the sugar away and to form the complex. So the science behind this is that if you remove the sugar and keep only the monosaccharide, which is close to the protein, then you are able to expose the highly conserved epitope and still make sure that the protein is still intact. And then we studied in more details, and we found that actually sugar can have a hydrophobic interaction with the nearby phenylalanine residue there, and that is about 1 kilocalorie in terms of binding energy. And once the sugar interact with the phenylalanine ring, it changes the orientation of the aromatic ring, and then followed by a domino effect. So we'll have a more hydrophobic interaction. And that is the reason for the stabilization of the monoglycosylated glycoprotein. So to have a good vaccine, you need to have not only memory B cell induction, but also memory CD8 T cell response. And in terms of antibody, I think other than binding to the target, and the antibody also needs to have a very good killing activity. And that is mediated by the Fc moiety of the antibody, and the Fc moiety of the antibody is also glycosylated. So you need to optimize the glycosylation to generate the antibody with the effector function we call ADCC, ADCP, and also the vaccinal effect. So then the next step is, can we apply this strategy to mRNA vaccine? And of course, mRNA doesn't have sugar to remove. You need to change the genetic code to delete the glycosylation. So what we did is to delete the glycosylation site in the stem and also in the S2 domain. And what happened is that once you delete the glycosylation site, you simply change the genetic code from asparagine to glutamine to avoid glycosylation. Now you can see that the protection is much more effective and also broader. And also, more excitingly is that it induces a very, very strong CD8 T cell response. And I think the protection is mainly from the CD8 T cell response, and that is getting more and more important for the vaccination against any infectious disease. So here you can see that the immune response against not only the vaccination strain, but also many other mutants. And then we focus on the stem region. We simply eliminate the glycosylation site of the 6 glycoside, and that is on the right-hand side. And you can see that the immune response also is very broadly protective. It's not only against the vaccination strain, the wild type, that is the Wuhan strain, but also many, many other strains, including the Omicron and the coronavirus, which cause common cold, and also SARS and MERS. So this we think is very effective if you simply remove the sugar in the highly conserved region to make sure that the immune response is enhanced, and that will reach the broad protection purpose. And you can see that the titer for IgG1 is much higher. Then we know that mRNA delivery is a major problem, and so far there's no selective delivery of the mRNA vaccine to dendritic cell, the antigen-presenting cell. So we come up with a new bifunctional lipid nanoparticle approach. We designed 3 lipid here, and once you mix together with the mRNA, the mRNA is encapsulated inside the lipid nanoparticle, and that will stabilize the mRNA and also selectively deliver to dendritic cell after immunization. And you can see that on the left-hand side, the immune response is much, much stronger than the one without selective delivery. And on the right-hand side, the antibody response, you can see that the antibody titer against the vaccination strain is almost the same, but the titer against different variant is much, much higher. So that means once you expose the highly conserved epitope without glycosylation, then the immune response will be concentrated on the highly conserved epitope, and that is how it becomes broadly protective. So we also have a polymer designed to encapsulate the mRNA vaccine for selective delivery to the antigen-presenting cell. And here we use the aerial trimannose as a ligand for lysine on dendritic cell. And you can see that it's also very, very selective against the antigen-presenting dendritic cell. So this idea actually was started many, many years ago in 2009 when we worked on influenza vaccine. So we removed the sugar from hemagglutinin and trimer and to keep the protein as a monoglycosylated. And you can see that after immunization, the antibody response is higher and the protection is better than the one without deletion of the sugar on the surface. And there's an antibody, as you know, we call FI6. This antibody is universal. It's able to neutralize all type A influenza virus. And then the CR9114 is the antibody which is able to neutralize all type A and type B influenza viruses. But these antibodies have not been optimized. So we know these are humanized antibody because the protein in the FC moiety is already optimized, but the sugar is still not. It's still hamster. It's from CHO cell. So we optimize the sugar part. We humanize the sugar also to make a complete human antibody. And here you can see that indicated by red, the neutralization activity is much, much better, and the Chinese study is also much more protective. So we also have once you isolate the antibody which target the highly conserved epitope, you can turn that antibody into a universal antibody with a much more effective effector function there. And this is just to summarize the way to optimize antibody. You can engineer the protein sequence and also the sugar sequence to completely human-like structure, and then to optimize the effector function of the antibody. And now, in terms of manufacture, of course, everybody knows that the current manufactured antibody is based on cell culture, and we can use CRISPR to change the glycosylation pathway to enrich the glycoform you want and to make the, for example, in this case, the universal antibody, which is much more effective than the antibody originally isolated. So we engineer the protein sequence and also the sugar sequence through CRISPR, and you can make the antibody with the enriched glycoform you want to optimize the effector function. So what about mRNA? The mRNA vaccine for influenza virus, and we know the FDA recommend 3 strains now, H1N1, H3N2, and one type B from Victoria. So we use the same approach. We simply, this is just preliminary result. We take out the glycoside, the 5 glycoside in the stem, indicated in blue, and then we use that as a vaccine for immunization. You can see that the immune response is almost the same as the fully glycosylated one, but the efficacy against other mutants is much, much more effective. You can see that here, not only against H1N1, but also against H5N1, as indicated on the right-hand side in the bottom. So I think the engineering of the glycoside at the mRNA level or at the protein level, you can prepare the vaccine with a much more effectiveness against different type of variants. And this technology was selected as one of the top 10 emerging technology by IUPAC last year, and just want to share with you about this. So thank you very much for your attention. Yeah.